Yeah, we're in business. Okay. Okay, thanks, Sean. Okie doke. All right, now we'll get to vegetative cells and spores. Okay. Because of the, microbiology is like fashion. Women's skirts go up and down each year and what have you. And, with the anthrax attacks in 2002, it changed our perspective on things. I mean, basically, I like spores. I work with anthrax, so I'm very into spores. But this is not the reason that this title is here. I don't want you to get the wrong impression. I did mention this in another lecture. Most bacteria do not make spores. Okay? And because most bacteria do not make spores, we almost never use the term vegetative cell. Okay? Bacteria that are growing in a regular fashion and growing fast, they are vegetative. But the only reason you would use the word vegetative, that in my experience anyway, is because of the fact you're trying to differentiate a fast-growing bacterial cell from a dormant spore form. Okay, so the emphasis here is just including both, but that's not. Don't get carried away and think about all bacteria make spores. They don't. Most bacteria don't make spores, and we don't use that term vegetative unless we're talking about spore-forming bacteria such as Clostridia and bacilli. Okay, and I won't be covering the bacilli, but I will be covering the Clostridia. Okay, again, this is not just a shopping list. There's some logic as to why these terms are up here, trying to give you an overview of what we're going to be doing. I gave you a brief overview in an earlier lecture of the cell envelope. And I said to you that I'm going to show you the same slides again, but I'm really going to get into more detail. Okay. Okay. You haven't missed much. You just missed the overview. All right, so what we're going to do here is I'm going to explain what we're going to be doing here. Okay. First of all, a cell envelope refers to all the layers of a cell that are outside the cytoplasm. Okay, so it would include the cell membrane, the cell wall, and an outer membrane if one is present. You don't see the word cell envelope used an awful lot, quite honestly. What you do see used a lot is cell wall, cell membrane, and outer membrane. And the only reason I do this in this lecture is because people often use the word, even microbiologists use the word cell wall wrongly. And I want to basically explain correctly what we're talking about here. Okay, so cell wall, cell membrane, outer membrane. Within the cell envelope layer, there could also be the peptidoglycan layer, so there could actually be as many as 4 layers in the case of a gram-negative organism, and there could be as many as 3. There will be 3 usually in a gram-positive organism. There are unique things that are present either in gram-negative bacteria or gram-positive bacteria. Now, as I said, I'll just mention it again briefly, those of you who just got here. The title of this lecture is Vegetative Cells and Spores. Most bacteria don't make spores. We don't usually use the word vegetative unless we're talking about spore-forming bacteria, because vegetative bacteria are just ordinary bacteria that are just growing and spores are quite unique biochemically. We are going to discuss in detail the structure of an endospore and to show how it's quite different from a regular cell. But most bacteria, usually the major species, the major genera that cause human disease are the Clostridia and the bacilli. And they are the only ones. I think they're referred to as a spore-like body. I think it's in chlamydia, but I'm not going to go any further with that. But as regards truly defined spores, they're generally in the gram-positive organisms, bacilli and Clostridia. And we're going to talk about cell division, and we're going to emphasize the difference. That's why it's at the end. We're going to emphasize the difference between what happens in regular replication as opposed to what happens in sporulation. Okay, topics. Structure of vegetative cell envelopes. As I say, probably as the years go by and we forget about the anthrax attacks of 2002, I'll probably drop the word vegetative from this topic again. But anyway, as it stands at the moment, vegetative cells, just regular cell envelopes, and bacteria that either don't make spores or the fast-growing forms of those that do make spores. There are unique cell envelopes, biosynthesis of cell wall constituents. We're going to talk about endospores and how they're different, and we're going to talk about cell division and what's different in sporulation versus regular cell replication. And as I already said just now, to emphasize again, the cell membrane plus the cell wall plus the outer membrane, if it's present, makes up the cell envelope. Now, the cell wall. Let's talk about the cell wall. Again, as I said the other day, the wall is a rigid structure, but it's not. But as I want to point out, it is just like this wall in terms of rigidity, but it's a molecule. It's not a hunk of wood. The whole bacterial cell is made up of, the shape comes from a single molecule of peptidoglycan, which can be billions in molecular weight. Billions. Not thousands, not like a protein, not millions, billions in molecular weight. It's a huge thing. One single molecule making up the entire wall of the bacterial cell. And its backbone is referred to as peptidoglycan. Those of you and you all have had anatomy courses and other courses and what have you. I did not say proteoglycan. I said peptidoglycan. It sounds very similar. A peptidoglycan has got absolutely nothing whatsoever to do with a proteoglycan. A peptidoglycan specifically is the cell wall structure of a bacterial cell. It has a very unique structure, which we will discuss. Depending on whether you have a gram-negative or a gram-positive organism, there can be unique, different structures can be attached to this peptidoglycan layer. So you need to know the differences, not just in terms of the thickness of the peptidoglycan that I discussed earlier, and the structures of the peptidoglycan, which is much the same fundamentally between gram-positive and gram-negative bacteria. But you need to also understand the unique structures that are present in the gram-negatives and gram-positives and why they are, what they're doing and why they're there. So again, as I said in an earlier lecture, when I discuss E. coli, not now, there's going to be two parts of this course, basic and clinical, okay? And I have a summary table which I'll give you in the 6th lecture. For quiz number 1, you're not required to learn all the bacterial species because you haven't had all the bacterial species. You won't have had the clinical part of the course. So it would be ridiculous to ask you to sit down and memorize it. But you will have had, in the clinical part, we will go through every organism that's important to human disease, which there aren't that many, fortunately. And again, if you see an organism gram-negative, and you know this lecture well and understand it, you won't have to go, okay, I know that E. coli is gram-negative, I know that Vibrio cholerae is gram-negative, I know that Bacillus enterosus is gram-positive, etc. If you know this, you don't have to sit and say, well, does it have a lipopolysaccharide or not? Because you know from this lecture that if it's gram-negative, that's what you're going to find. Okay? So that's the point. Pull together the basic part of the course with the clinical part when you get there, because you're making life hard for yourself if you don't. Okay? All right, so let's go through this now. Gram-negatives, gram-positives, etc., etc. Bacterial cell envelopes. Gram-positive or negative, it correlates with structure, and I've just chosen to not go into the reasons why because I think it's beyond the scope of this course. So that part we will memorize. The gram stain usually correlates with the structure. And the key word was usually. It doesn't always. To take an example from that horrible term we used in the previous lecture, autolysis. If a bacterial cell was to autolyse, by definition, one of the things we're going to be getting into, and more so in the next lecture, is the synthesis of peptidoglycan, and we're going to get into how antibiotics act. If an organism chews itself up through autolysis, it particularly affects the structure of the cell wall. So if an organism is chewed up, a gram-positive organism will now become it'll lose the diet more easily, as an example. So an organism that is autolytic, that would normally be gram-positive, could appear gram-negative. Why do you care? You care because if you happen to be a resident or a infectious disease resident or postdoc, and you're looking down a scope at some samples from a patient who's might have meningitis, and you want to know whether the person's got Haemophilus infection or has got a pneumococcal infection, both of them are cocci. And the only way you can tell them apart by looking at them is whether they're gram-negative or gram-positive. And if you don't know that the pneumococcus, as I will discuss later, is autolytic and therefore could appear gram-negative, you could totally confuse whether you've got a Haemophilus or whether you've got a pneumococcus. I'm not asking you to memorize that now. I'm saying to you, this is an example, okay? So this is why we care about these sorts of things. So the key word here was usually, not always, does a gram-positive structure correlate with the gram stain. But it usually does. Okay, another example. This is the sort of onion skin model, where we basically just reduced the bacterial cell envelope to lines and layers, the peptidoglycan and the plasma or cytoplasmic or cell membrane in the case of gram-positive bacteria, the gram-negative bacterial being sort of 3 layers with the peptidoglycan or cell wall in the inside. Okay, peptidoglycan. Some people really synonymously say cell wall and peptidoglycan because it really is the cell wall. It's the molecule that, go ahead. Yes, yes. That's why I've made a point of saying all these terms, because I don't want anybody to say, I want you to remember, whatever term you see, you recognize it. And if one that I don't say, you recognize it because you understand the lecture. Okay, but yes, absolutely. Plasma membrane, cell membrane, cytoplasmic membrane. If you can think of any other form you've heard somewhere else, that applies too. Okay, the peptidoglycan has only got 2 names usually, peptidoglycan and murine, which is in the handout. Peptidoglycan is the most commonly used term. And I'll show you why in a second. It's a single molecule, highly cross-linked, because the pieces of the peptidoglycan are pretty small. And I've already said a peptidoglycan molecule is the whole size of a bacterial cell. It's the framework. And the pieces are held together by the cross-links. You degrade the cross-links, the cell falls apart, the osmotic pressure pushes on the membranes, it lyses the cells. And that's one of the major mechanisms of antibiotics, which I'll talk about in the next lecture. It provides rigidity. Now, there was a good question that was asked by some, I think it was in the front here the other day, about permeability. Okay, I don't want you to, that's why I banged on the wall the second time this time. I meant to get there. Don't get confused when I talk about walls as being sort of solid. The peptidoglycan is a molecule. It's got holes in it. Okay? Things can get through the peptidoglycan. It's not really a permeability barrier. Okay? In the case of gram-negative bacteria, it's not a barrier at all because it's only 1 layer thick. In the case of a gram-positive bacterium, it's somewhat of a barrier because things of about 100,000 molecular weight can get through, but not beyond. But it isn't really a barrier like the true barrier in, as we said earlier, which we're going to say again today, is the outer membrane in gram-negative bacteria. Cell wall should not be thought of as a barrier. It's there to provide rigidity, strength, and that's what a wall is, rigidity and strength, to keep the loose, fluid, phospholipid-containing membrane underneath from the osmotic pressure pushing outwards on it and causing it to fall apart. That's the primary function of the cell wall. There are other functions, but that's its primary function. Okay, why do we call it peptidoglycan? We call it peptidoglycan because it has 2 components, a glycan backbone, and it has two peptide components. Sometimes there'll be 1, sometimes there's 2. I'll show you what I mean by that in a second. But peptidoglycan, a peptide part, an amino acid containing part, and a glycan part, a sugar containing part. Now, why did I make a big fuss of the fact this is not a proteoglycan? Well, for a number of reasons. First of all, it isn't a protein at all. It's a peptidoglycan. It's not made on a ribosome. We're going to talk about the structure of synthesis. The molecule's got nothing whatsoever to do with proteins, in fact. First of all, let me say, muramic acid, basically the only place in nature that muramic acid has been described, and a lot of those studies have come out of my lab, but the only place you find it in nature is in the bacterial cell wall. You don't find it elsewhere in nature. You don't find proteins contain muramic acid. Human proteoglycans, for example, do not contain muramic acid. Secondly, the amino acids, despite what NASA said some years ago, the amino acids are found on this planet. They are found in bacteria. In fact, almost every bacterial cell on the planet, everyone that has a wall, has the amino acids. Proteins do not contain the amino acids. There's chirality in life on this planet, and ribosomes are designed to put together L-amino acids into proteins. So as soon as you see the amino acids, you're saying, okay, this couldn't be made on a ribosome. Okay? So this has got nothing to do with proteins. It's got nothing. And in fact, the sugars are unique. There's other compounds like diaminopimelic acid, which is almost never found in nature also, except in the peptidoglycan. This is a totally unique molecule. structurally, component-wise, any way you want to look at it. We do occasionally see D-amino acids in people. For example, as people age, you see some natural chirality change as proteins are broken down into amino acids, and the amino acids may be naturally racemized into D-amino acids. We see low concentrations, but generally speaking, naturally produced, metabolized. You don't see D-amino acids in nature, except in rare places like the peptidoglycan. So when you see D-amino acids and you think about microbiology, immediately you're saying, oh yeah, peptidoglycan. Okay, now, the structure of, there is a famous review published about when this sort of thing was popular about 30 years ago. It's about that thick by a guy called Krause. Krause was one of the people, I've forgotten the other name, Schleifer, Karl Schleifer. Every page has a different peptidoglycan structure on it. Again, if those of you that want to go down the library, it's there, get hold of it. You can memorize every page has a different structure on it. I think it's absolutely absurd. What I want you to understand is what this peptidoglycan is and how it differs among different organisms, not to memorize. You need some of the important parts, but we don't want to go through peptidoglycan after peptidoglycan after peptidoglycan. And I won't show the structure again after the next couple of lectures. When we talk about antibiotics as well, we're going to discuss it. All right. First of all, the peptidoglycans generally consist of a subunit of 5 amino acids: L-alanine, D-glutamic acid, L-lysine, and there are usually 2 amino acids on the end, the D-alanines. The reason we focus on knowing this in detail is because we cannot understand the function of important antibiotics such as penicillin if we don't know anything about the structure of peptidoglycan. Because that's where almost half of the antibiotics that you'll ever give to a patient, where they're working and acting. And it's why we have a pharmaceutical industry that does good chunks of it, do nothing but make new antibiotics every year to try and make different variations to act on different bugs. So we need to get an idea of what we're dealing with. Okay, first of all, the peptidoglycan. The backbone, so the side chain is made of these 5 and sometimes 4 amino acids. The backbone is made of this boring sugar, glucosamine, which is found everywhere, including human proteoglycans. But this good friend of mine, muramic acid, is only found in bacteria. Okay, so that's C-muramic acid, that's peptidoglycan. Now, there can be some variation in this structure. It's usually LD, LD, and then an extra thing on the end. But it differs somewhat between, that's why it says lysine or diaminopimelic acid. Gram-positives generally have lysine, and the key word there was generally, it didn't say always. Gram-positives generally have lysines. Diaminopimelic acid is often found in gram-negatives, and that's one reason why we like to know this. The sequence can vary a bit. But the reason there's whole reviews and books just showing different structures is because the peptidoglycan chains are held together by a cross bridge. As I've said already, if you don't have that cross bridge, and what you've got is tiny molecules, just a few sugars and amino acids in length, you don't have a cell wall, you don't have live bacteria. That bridge is what holds it all together. Now, in some cases it can be a single peptide bond. In some cases it can be as many as 5 amino acids. In fact, some cases it can be the same 5 amino acids that we see here. From the point of view of understanding what peptidoglycan does and how its importance in, for example, in our use of antibiotics, the variation here isn't really relevant because most of what we care about is this fact that this bond is either there or it isn't there. And what we care about are these 2 amino acids because they're involved in the process. So I will go through, when I get to the antibiotic lecture, the next one, I will show you a mechanism for a number of different antibiotics. And I have different slides for each antibiotic. And if you read the words and you don't look at the pictures, it sounds like I'm saying the same thing every time. If you look at the pictures, you'll see, no, I'm not. There's something different in each slide. It all relates to these 2 amino acids here and that bridge. Okay? All right, so we'll get more into that later. Okay, gram-positive cell envelopes. The gram-positive cell envelope usually has linked to it, it can have neutral polysaccharides, but it often has teichoic acids or tichuronic acids. And the neutral polysaccharides are not emphasized usually because people find it boring. And so what they do find interesting is what these teichoic acids and teichuronic acids do. And so this is more emphasized. A teichoic acid is essentially, well, a simple way to think about it is it's an analog of the chain of DNA. It's not a carbohydrate. These are molecules that are based on carbohydrates, but they're not carbohydrates. The difference between a carbohydrate, I remind you from your biochemistry, and DNA is that carbohydrates are held together by glycosidic linkages, whereas DNA chains are held together by phosphodiester bridges. Again, I'm not going to make a big deal about biochemistry here, but I'll just jog your memory so that you see where I'm going with this. All right, this is not a carbohydrate. This is a teichoic acid. It's a polymer held together with lots of phosphorus. Usually, there are 3 or 5 carbon sugars, ribitol or glycerol, in the backbone. I'm not going to make a big deal about the exact structures. I don't think it's not where I'm going. What I am going to do is emphasize the difference between a teichoic and a teichuronic acid and why we care. A teichuronic acid so this molecule is highly negatively charged because it's got a bunch of phosphate groups attached. A teichuronic acid, by definition, an acid is a negatively charged ion. A teichuronic acid must also be negatively charged. It indeed has a lot of carboxyl groups there. It is actually a carbohydrate. It does not have these phosphodiester bridges that you see in a teichoic acid. Now, there's no phosphorus, but it does have glucuronic acids. The glucuronic acid is substituting for the phosphorus. And what happens is, if you grow a gram, and it has to be gram-positive we're talking about here again, because it's emphasized in the handout, because we don't find teichoic acids or teichuronic acids in gram-negative organisms. So if you ever see teichoic acid, you ever see teichuronic acid, you immediately know you're talking about a gram-positive, not a gram-negative organism. Okay, so what happens with these gram-positive organisms, if they do this, they grow, when they're grown in the presence of lots of phosphorus, they will make teichoic acids preferentially. If they're put in nutrient depletion conditions where there isn't a lot of phosphorus around, say there's no RNA or DNA to chew up and grow on for whatever reason, they don't die. They're smarter than that. They preferentially use their phosphorus to make DNA and RNA. And instead what they do is they make teichuronic acids. Now, what you should be saying to yourself, he keeps emphasizing charge over and over again here, but he hasn't told me why. You might also say, I don't care. But if you do care, the reason is because these molecules are involved in ion uptake, not I-R-O-N, ion, I-O-N, metal ion uptake. They're involved in taking up positively charged metal ions from the environment and serving as a metal ion sink, so that bacteria can pull these metal ions up into their cell walls so they can use it for metabolism. Okay? And if they don't have a lot of phosphorus, they can't make teichoic acids. So over the years, they've developed, evolution's come in, and they decided, hey, it's a pretty good idea to make these teichuronic acids, because if they don't, they have no way to grow. So this is in low phosphorus concentration. This is in high phosphorus concentration, and that's the reason. Okay? Teichoic acid, teichuronic acid switch. These are covalently bound to the peptidoglycan. I shall emphasize that word a lot, covalent and non-covalent. Not so much in the slides, but in the handout. Okay, these again are covalently attached to the peptidoglycan, the teichoic acids or the teichuronic acids. So again, metal ion uptake, and I'm going to use that ghastly word again, autolysis, which is why I made the previous slide, which I couldn't remember this morning. Yeah, go ahead. Right. I'm sorry? Well, you tell me. What do you think? Okay. Okay, metal ions are positively charged, all right? So what would you need to take up a positive charge? So what would you think a teichoic and a teichuronic acid would be? But I'll ask you a question. What do you think they would be? Thank you. Okay. Yes, I was saying that. Okay. I'm saying again that the teichoic acid, okay, I'll explain. But this is how I want you to think when you're not with me. Okay? Because that's how the lecture is designed. It's put together, so I hope it's obvious. And what I'm trying to do here, I will answer your question, it's a good question. But what I'm saying to you is that when you go home, read the handout, look at the slides, and if it doesn't make sense, it's there somewhere, hopefully. If it's not there, let me know and I'll make another slide for the next group. You'll lose out, but the next group hopefully will benefit. Okay. All right. So coming back to your question. Yes, again, bacteria that are grown under low phosphate conditions cannot make a teichoic acid. All right? They will make a tichuronic acid under low phosphate conditions. Under high phosphate conditions, they will make a teichoic acid. Okay? Is that clear? Okay?
But wasn't phosphate the positive ion and negative?
If I said positive, then I screwed up. I meant phosphate to be a negative ion. So did I say positive? Well, I don't know. If I said it, then I'm taped. I'm screwed, you know. But phosphate is negatively charged. So it's involved, the negatively charged teichoic acid is involved in taking up positive metal ions. Okay? If you have a problem, come and see me after class and we'll go over it some more, okay? All right. Okay, I realize some of you are history majors in this class, but no offense. But anyway, okay. All right. We get, so let's move on. But anyway, I'm very happy. If any of this stuff is not clear, seriously, folks, come to me after class, or my office is there. If I'm busy, it's very simple. I'll answer the door, have a bell, and unfortunately lock it, and I'll say, go away. Come back later. But I will make a time you can come back and see me, okay? Most of the time I'll say, come on in and we'll talk, okay? All right. Teichoic, tichuronic acid involved in positively ion metal ion uptake. And now we're talking about autolysis, okay? All right. Again, autolysis is not what it says. Auto means self, lytic means digest, self-digestive. Bacteria, unlike medical students, are not suicidal. Okay? They don't want to kill themselves. Autolysis, unfortunately, is a byproduct of cell wall metabolism, which we will get into in a little bit. Autolysis, what's going on is you've got this huge massive structure, the peptidoglycan. And it's held together by these cross bridges that I've been talking about. The only way you can make new peptidoglycan is to cut a piece out and make a hole. You make a hole, you can insert something. Now, if the process goes awry, what's going to happen is, so in fact, peptidoglycan or cell wall biosynthesis consists of two parts. Self-digestion to make a hole, and then actual synthesis to put another piece in, a larger piece in, for example, to grow. So, what happens is, if the autolytic process is not well controlled, for example, if you give an organism an antibiotic that's going to inhibit cell wall biosynthesis, then the autolytic enzymes will kick in, and they will contribute to the role of the antibiotic in killing the bacteria. Okay? So autolysis, is not there for suicide. It's there as part of the synthesis of cell walls. And in the case of the pneumococcus that I mentioned earlier, it unfortunately, whatever reason, it's so poorly controlled that the organism actually, when you grow it in the laboratory, it actually does digest itself. Whether it has a role in human disease, who knows? It is interesting that the pneumococcus is on the top 10 killers, not infectious diseases, but still the top 10 killers in this country. One of them is pneumococcal infection. So, do you have a question? I'm sorry, you just touching your hair. All right. Okay, so and the reason, what happens is that these teichoic and not so much the tichuronic acid, what happens is that the teichoic acids are believed to be involved in the control process. They actually tell the enzymes involved in autolysis where to go. Presumably there's some sort of stereochemistry going on, stoichiometry, that in the bacterial cell wall, the teichoic acid is put in a certain place to say to the autolytic, hey guys, come over here, you've got to do something. So that's why we're talking about the teichoic acid. They direct the autolytic enzymes to basically make sure that we punch holes where we should punch them and carry out peptidoglycan biosynthesis in the appropriate fashion. Now, don't be confused by this term, and I do believe Murray is still confused by it. I did at one point waste my time contacting the editor and tried to explain it, but he's a clinical microbiologist, so I don't think he got it. Let me explain to you so you understand. Most microbiologists do know this. Lipoteichoic acids are not the same thing as teichoic acids. Yes, of course, they structurally have some similarity, and I won't go into a lot of details. They do, because that's why they're called teichoic acids. But the fact that it's got the word lipid there, as you would expect, the fatty acids attached to, to this teichoic acid, which makes this molecule quite hydrophobic. The difference comes in is that this is not found covalently bound to the peptidoglycan. It is found in the cell membrane. And it also may well be involved, and we can get in, I think I may even have a slide on this later on. But the lipoteichoic acids are also involved. Actually, I do have a slide on this later on. The lipoteichoic acids are involved in keeping the autolysins away from the cell wall. Because, again, the cell wall is one layer, the cell membrane's underneath, and if the lipoteichoic acids are down here, the autolytic enzymes are bound to them, they can't get at the peptidoglycan with the teichoic acids up here. Okay? So they're structurally related, and they do relate to autolysis, but they have different functions. And the point is that, again, gram-positive. You see the word teichoic acid, you know you're talking about a gram-positive. Not all gram-positive bacteria have lipoteichoic acids. We will mention some cases where it's important later on. But I want you to understand that the lipoteichoic acid is a cell membrane molecule. It can obviously get out of the cell membrane in certain circumstances, but it's primarily a cell membrane molecule. A teichoic acid is covalently bound to the peptidoglycan. Okay, so those are so far we've discussed the unique gram-positive features, the teichoic acids, the teichuronic acids, the lipoteichoic acids. We know what they do. We know why they're there. So let's take a look at the gram-positive cell envelope. And it is a lot simpler than the gram-negative cell envelope. Now, unlike the last time we showed you this slide, same slide, you know a lot more. All right, again, cytoplasm, cytoplasmic membrane, whatever you want to call it, it's there. It's a regular. The reason I say this because it's just no big deal. It's nothing. It's just the same. Any form of life on this planet has got this sort of thing, and that will contrast to the outer membrane I'll get to next. And so there's no reason to think about a bacterial. A lot of times people say cells, and then they say bacteria. No, excuse me, they are cells. Okay, they're just cells with a wall. Okay, so of course they've got a cell membrane. There's nothing special, and those of you who've had classes that you all had in the past in biology and biochemistry and what have you, you know what a membrane is. Nothing special about this membrane. The peptidoglycan is unique, and we show here the lipoteichoic acids and the teichoic acids. The teichoic acid, that dash, is there meant to imply that they're covalently attached to one another. And you'll see that the teichoic acid is bound by this little red dot, which is, of course, a lipid, because we know that membranes are hydrophobic, very fat-loving. We know that things that are fat-loving or hydrophobic will be found better than membranes, so we don't have to think too hard about this to know that that has to be the membrane part, and that's the carbohydrate part, or the phosphate-containing carbohydrate part of the lipoteichoic acid. Right? So those are the unique features of gram-positives. And I stress again, any time we go into the clinical part and we use these terms, I'm not going to go over it again every time. We don't have time. Go back and look at this lecture, okay? All right. The peptidoglycan. In the case of the gram-negative bacteria, the lipoprotein is covalently bound to the peptidoglycan. It substitutes for the teichoic acids and the teichuronic acids, and we don't find lipoteichoic acids in the membranes of gram-negative bacteria. The function, again, we see the word lipid. It's there for a reason. Anytime you're going to see a lipid, you know, again, it's hydrophobic. You know that it's got to be something to do with a membrane, or probably something to do with a membrane. So what happens is the Braun lipoprotein, named after Volkmar Braun, who published an article on this in around the early 70s, called the Braun lipoprotein. I have to discover something, have your name after it, isn't it? Yeah. Anyway, the Braun lipoprotein basically binds cell walls to the outer membrane. I'll show that in the next slide. The other thing I also mentioned that's unique is the outer membrane, and I will stress it again. The outer membrane here is, now, when we say, somebody asked a good question earlier about plasma membranes versus cell membranes and cytoplasmic membranes. And as I said, these are boring old things. But when I say outer membrane, and you would immediately say to yourself, if I said inner membrane, would that be a gram-positive or a gram-negative? Well, I wouldn't use the word inner membrane ever when I'm talking about a gram-positive, because there is only one membrane. And of course it's not inner, because there's nothing outside it. Okay? So again, when you're looking through this stuff, look at these distinctions, okay, and what have you. So the point is that there is an outer membrane here because there is a cytoplasmic membrane, which is the inner membrane in gram-negative bacteria. And what's unique about this membrane is the lipopolysaccharide. Yes, there's boring old phospholipids there, there's boring old proteins there, and there are unique proteins called porins. Now let's look at this slide, actually. We'll go a little bit about these things. Now, I go back and forth on this. I sometimes show nothing, sometimes I show... I used to show the whole hog, and I've got rid of that. I want you to just basically get the basic understanding about what a lipopolysaccharide is. I would remind you that lipopolysaccharides are different in just about every organism in one part of themselves. But, again, just like we said about the peptidoglycan, we could spend, a week just looking at structure after structure after structure after structure, and that's not what I want to do. What I want you to do is get the idea of what is a lipopolysaccharide. First of all, it has several components. It has these fatty acid parts. Now, most of the time I say fatty acids, I don't mention anything. These things are different. They're called beta-hydroxy fatty acids, and this is not the only place they're found in nature, but it's one of the few, let me put it this way, this huge amount of it is the only place in nature that you find huge amounts of beta-hydroxy fatty acids. But the point is they're characteristic of the lipopolysaccharides. That's why I've emphasized it. The lipid A region, and these can vary a little bit in structure between one organism and another, but they always have hydroxy fatty acids. And you'll see as we go from the inside to the outside of the lipopolysaccharide, the amount of variation increases. So we've got basically always got these hydroxy fatty acids, different chain lengths and what have you, but they're always there. The lipid A part is always there, and that is a glucosamine disaccharide, and there can be some differences in phosphorylation and other things. But basically, a lipid A is a lipid A. Then we get to the core region. Again, we're moving from the outside. Things are starting to, still much the same we said so far. They have things called heptoses, 7 carbon sugars. You don't find 7 carbon sugars very commonly in nature in structural things. It's usually that boring old sugar in the Krebs cycle we keep talking about over and over again. Glucose, 6 carbon sugars. This is a 7 carbon sugar. And there's this other unpronounceable name, ketodeoxyoctonic acid and other versions thereof, which is also a molecule that's generally only found in the core region of lipopolysaccharides. Now, at the end of the molecule, there can be a lot of variation, and it can differ between different species and closely related species. Now, you might say to yourself, okay, that was nice, but what was that all about? Okay, what it's all about is this. This is one of the things that's going to kill your patients. And you can choose to just say, well, they died. Or you can choose to say, okay, I know why they're dying. And it's called septic shock. And lipid A this is another name for this, is endotoxin. And in my pathogenesis lecture, I'm going to go back to the word endotoxin, but we're talking about bacterial structure today, different purpose. Okay, so this thing is what kills, probably a couple 100,000 people a year, every year in the United States, and we still don't have an effective way to deal with it, even though people have tried very hard over the years to find a way to deal with it. Okay, so this is that, and that's why it's so important that it's invariant, or not invariant, but very similar among one organism and another, because if you can get gram-negative infections, you're going to have a good possibility of getting septic shock and endotoxemia. The reason this is important on the outside is because it's used clinically to differentiate, for example, among different strains that might be involved in an epidemic. And we'll talk, for example, those who've heard about enterohemorrhagic E. coli and, hamburger disease. I would call it, it's not a strict term, hamburger disease, but you remember it was associated with hamburgers. And basically, there's variations in this part of the molecule, which is referred to as the O antigen. Okay, so we will discuss, particularly as regards the Enterobacteriaceae later on, we will discuss lipopolysaccharides. We will discuss lipopolysaccharide in the pathogenesis lecture. What we won't do is ever show this slide again, but we're going to assume that you remember it. Okay, that's the lipopolysaccharides, unique to gram-negative bacteria. Okay, so let's look at now with a little more detail than we did in our first lecture. Let's look at the gram-negative cell envelope. Again, cytoplasm, cytoplasmic membrane, usually referred to as a single layer thick in gram-negative bacteria, but here's where the interesting stuff starts to happen. First of all, There's brawn lipoprotein showing you here covalently bound to the peptidoglycan. There's some of it is actually found in the outer membrane, loosely attached there. Most of it's covalently bound to the peptidoglycan. But the issue is that this peptidoglycan, this brawn lipoprotein, when it's attached to the peptidoglycan, it's what holds and anchors the peptidoglycan, anchors the outer membrane in place. This is a fluid thing, as I've said before, and it could just sort of float around and, disappear if it wasn't held in place. And that's what the brawn lipoprotein does. In addition, I keep mentioning about permeability barriers. As I mentioned already, this might have a little bit of a function being quite thick in gram-positive bacteria, but there's essentially nothing there. It's a single layer thick in the case of gram-negative bacteria. This is the permeability barrier. And bacteria are smart. They have developed over the years things called porins, because what happens is, you'll notice here, this is unusual. You'll see these little chains hanging out. These are carbohydrates. Carbohydrates are hydrophilic, water-loving. That means if a dye, for example, a hydrophobic dye or something that you might find, a bile salt, for example, in the gut, comes into contact with a gram-negative organism, it cannot get at this underlying lipid, phospholipid part, or it cannot get at the cell membrane, because the lipopolysaccharide has this carbohydrate chains coating the surface. In addition, however, if it was a barrier as such, if it was a totally stupid barrier, then that would mean nothing could get through. If nothing could get through, well, how could the bacteria survive? It had nothing to grow on. So it has to have a way of selectively getting things through this outer membrane. And one of the ways it does it is through these porins. In the case of the porins, these are protein channels which are designed to only let small molecules pass through, like oligosaccharides, for example. In other words, the gram-negative organism secretes stuff, chews it up, and then it can get in through these porins. Okay, so this relates to the first lecture we had on digestion. Okay, so the unique things here are the brawn lipoprotein, which usually covalently bound to the peptidoglycan, but always is characteristic of a gram-negative organism. You will not find lipopolysaccharides anywhere except in gram-negative bacteria, and you won't find these porins because there's no outer membrane in anything but a gram-negative bacterium. Okay, this was a new slide that I put together a couple of years ago which is very simplistic, but I think it needs to be done. We will have a lecture on the mycobacteria and corynebacteria later. And the thing is these are different. And the mycobacteria, there's a specific stain that is used diagnostically called the acid-fast or Zil-Nielsen stain, which is used specifically to make a diagnosis of TB or tuberculosis. And so this relates to understanding what's different about mycobacteria. And we will have a few slides on that, quite a bit more when we get into the mycobacteria lecture. But this time I want you to understand what's different. First of all, people say, well, is it gram-negative or is it gram-positive? Well, the answer is neither. It's got some features of a gram-negative organism and it's got some features of a gram-positive, but it's totally different to both. First of all, again, as always, it will have a cell membrane. It has a thick peptidoglycan layer. But in this instance, it does not have brawn lipoproteins. It does not have teichoic acids. It does not have neutral polysaccharide. Actually, it does have a neutral polysaccharide. But it's got what's the arabinogalactan. But what it does have is these weird long-chain fatty acids called mycolic acids, which is basically, I don't know which way around it was, whether the mycobacteria were named after the mycolic acids or vice versa. But anyway, this is what makes a mycobacteria or a corynebacteria or nocardia, which we'll go into detail with those names and what they are later on. This is what makes them unique. They have mycolic acids covalently linked to their peptidoglycan and a neutral sugar, arabinogalactans, after that. They do have, in a sense, for a long time people, and I'm just for simplicity putting it this way, they in a sense do have an outer membrane because they have they have a waxy layer, which is, and I purposely drew it a different color to the cell membrane because I want to make clear it's not a regular membrane. It's got all sorts of weird things there which are so complicated that chemists spend their lives working out their structures, and I'm not joking in that one. Mycolic acid lipids that are found in this layer. Okay, so acid-fast bacteria are different. And again, as I said in the earlier lecture, these two I'm referring here to mycobacteria and mycolic acids. This has got absolutely nothing whatsoever to do with mycoplasmas that I mentioned in an earlier lecture, and which I think Dr. Gaffar, Dr. Mayer will be lecturing on. Okay, so this is mycobacteria, not mycoplasma. We'll talk a little bit about synthesis, and we will go over this a lot. You got a question? Yeah, that's good.
For glycolic
I am
acid
too.
and acid fast
Yeah.
envelope are just sort of going together.
Yes, absolutely. Because it's what the, okay, let me give you the short version now, but I'll give you the long version afterwards, okay? All right, the short version is that this membrane is what makes these organisms acid fast. And there are different types of mycolic acids among the different organisms that are related to mycobacteria, which are the Mycobacteria, Corynebacteria, and Nocardia. I will have several slides just showing the differences among them, okay? So it's a good question, but just leave it for now. This particular lecture, what you need to know is that the mycolic acids are unique to the group of organisms related to mycobacteria. Okay? All right. Okay, peptidoglycan synthesis. All right. Now, this slide, it wouldn't be a bad thing if you don't get it because we're getting a bit late now. If we don't, have a look at it before tomorrow because this slide's going to come up in different forms about 6 times. It's not tomorrow, Thursday, I think, when my antibiotic lecture is. So let me go over it now briefly, but we will be going over it again because we're going to it's going to use it to see how the different antibiotics that we're interested in work. All right. Peptidoglycan, again, this is not proteoglycan, this is not protein synthesis. This has got nothing whatsoever to do with ribosomes because it's got the amino acids, among other things. And ribosomes don't have anything to do with the amino acids. Okay, in the cytoplasm, there are 3 stages in the synthesis of the cell wall. The first stage is in the cytoplasm, the sugars, which are labeled in red, and the amino acids, which are labeled in green, are put together, and the individual sugars and amino acids are made. Okay? We also get a subunit form. Now, people get confused by this word subunit, and I know that sometimes lecturers are tricky. Sometimes they don't mean to be tricky. It's just that words are used in different ways. Okay? And I try very hard. I don't see any reason to be tricky. But people get, the point is this is a subunit, and so is that. It's a word. Okay, so I'm going to try and use concepts and not just words here. Okay, so this is a subunit. These subunits, these 2 subunits are put together in the cytoplasm. Okay? But in the cell membrane, a bigger subunit is put together. Here we see this subunit and this subunit have been put together. This is the peptide side chain, and this would be, in this instance, it's not a direct amino acid or peptide bridge. It's a 5 amino acid or pentapeptide bridge. Okay, in this instance, the peptidoglycan gets attached to a lipid called undecaprenol or bactoprenol, which is also mentioned in the handout. So here, we've got stages. We've got 2 stages: individual sugars and amino acids, and there has to be something going on as regards synthesis because we know already that the muramic acid is unique to bacteria, and we know that the amino acids are unique. So they have to be making something unique there. They can't just be chewing up a hunk of cheese or bread. They've got to actually make this stuff because these are unique to bacteria. So there's metabolism making these unique components. We see small subunits put together and larger subunits are then put together in the membrane. The larger subunits are passed to the cell wall. And what happens in the cell wall, here you see now, the reason they're doing different colors is because this would be the original, the new sugars, and these are going to be the ones that are already there. This is the glycan backbone. And what you're going to see is that the new sugars are now attached and inserted into place in the old cell wall. At the same time, what you'll see is that the new piece of peptidoglycan came along, and yes, it does vary whether it has 5 or 4 amino acids, and this is why. And this we will again stress a lot in the antibiotic part. We see the new piece came along, and it had 5 amino acids attached. We also see that it had a subunit come along. But in order to form a peptide bond with the old piece of peptidoglycan, one of the amino acids, the D-alanine, had to be knocked off. Okay? So these are the stages, and this particular part here we're going to really emphasize when it comes to antibiotics. So that's why I've made a big fuss about the two D-alanines, not because I think you need to memorize whether peptidoglycan's got 2 amino acids sticking at the end of its chain, because you cannot understand how antibiotics work if you don't understand this structure and you don't understand how these 2 amino acids are sitting at the end. Okay? So take a look at that slide. I haven't made a big fuss about the lipopolysaccharide. There is actually no antibiotic that currently exists that actually is used to inhibit lipopolysaccharide biosynthesis. And one of these days when we have a large epidemic, the world and pharmaceutical industry will wake up and probably try and make one. But at this stage of the game, we don't have one. So the synthesis is similar to the peptidoglycan. So I won't be making a fuss about lipopolysaccharide biosynthesis. Okay, we're going to now discuss the spore. All right. Now, the spore is a modified gram-positive bacterial cell. It has a very unusual cell envelope because this spore does have an outer membrane. It's a hell of a lot more unusual than that. But this is just the beginning. Okay. In the case of the spore, yes, it's a bacterial cell. It does have a peptidoglycan layer. Spore folks like to give it a different name. Spore people like to name everything differently to confuse people. So they refer to it as a cortex. But it is a peptidoglycan, just like any other peptidoglycan. It is found between the two membranes. And it is less cross-linked, that is true. The major difference between the peptidoglycan of the spore and the regular bacterial cell is that the muramic acid is dehydrated. It's referred to as a lactam. It's lost water. And we will show the structure of a lactam in a different case Thursday as regards antibiotics. But anyway, that's mainly what's different. It's a little different, but basically it has that the peptidoglycan is much of a muchness anywhere you find it. Now let's look at why the spore is different. Okay. In regular replication, what happens with bacteria, they grow longitudinally. This shows the actual, not the moving, it shows the growth of the bacterial cell from this starting point. And bacterial cells will generally there's the bacterial cell here. Generally, when regular cells grow, they will start as a single cell and they will divide. And this shows the single cell growing away from this central point and forming two cells and a septum forming in between. What's different with a spore is that doesn't happen. What happens is it divides asymmetrically. It forms a small piece at the end, which is still a cell, and it's what will become the spore. Then what happens is, this is not phagocytosis, because we know that phagocytosis is carried out by amoeba and macrophages and neutrophils, and we know that you can only get phagocytosis when you've got a membrane. And this is a wall. So this is not phagocytosis. The mother cell grows around the daughter spore. And then what happens is you'll now see that there's what was before was blue and now is red. There are two membranes. One came from the mother cell, one came from the daughter cell, which will be the spore. So there are now two membranes. Now, just purposely, nature devised it this way to make exams difficult for medical students. What happens is, no, I am joking. Give it out the laugh. Okay, but anyway, what happens then is that the spore then forms additional layers in between the two membranes. And they are referred to as it forms additional layers outside the second layer. And that's referred to as the exosporium and the coat layers. So spores are totally different. They're totally different how they're made. They're totally different in their structures. Yes, they are gram-positive, but they do have an outer membrane. Let's take a look at what we end up with. Here we have, again, now we're referring to it as an inner membrane because there is an outer membrane. But we see a coat layer and we see an exosporium. Okay? So, that is the structure of the anthrax spore. And I've been quite interested in that because our group was the group that discovered what this exosporium was made of about 15 years ago. So I've got a little bit of interest in this. Okay. So anyway, that's the endospore. Now, just to show you how different these suckers look, and there was a lot of this stuff, in the last few years. You've probably all seen them in newspapers and websites and what have you. But we actually did take these pictures. These are the filaments of Bacillus anthracis, and you can see the individual vegetative cells. This is not a gram stain. This is a malachite green stain. And what you'll see is that the vegetative cells grow in chains. So if you ever see a blood sample, it has something looks like that, you've got to get worried. But when they sporulate, they don't look anything like this. And these are the individual. This is the same stain, but the spores, when they're produced, stain green. Actually, look a bit blue in this slide, but they're actually green. This is a malachite green stain. So, if you were to think to yourself, he showed me these pictures, they really, I think he's making a big fuss how different they are. Well, you don't have to know any biochemistry. All you got to do is just look under a microscope, And nobody on God's green earth could confuse something like that with something like that. So these endospores are very, very different. Okay? Okay, so that's what I think that's where we end up. Okay,